Hi, good morning, and welcome to our seminar. Thank you for joining us, and please tell us where you're joining from in the chat. We're happy to have you here. We're happy to have Emily as a speaker today. Dr. Emily Bellis earned her Bachelor of Science in Genetics and Biochemistry at Texas A&M in 2010. And she earned a PhD in integrated biology in 2017 from Oregon State University. She began working with plants as an SF fellow, postdoctoral fellow at Penn State, where her research focused on spatial coevolutionary genomics of serial parasitic plant interactions. I believe Emily worked in Striga. We talked about that the other day. In 2019, she returned to her home state of Arkansas and was an assistant professor of bioinformatics in the Department of Computer Science at Arkansas State University until this year, May 2023. She also served as Associate Director of the Center for No Boundary Thinking, leading several initiatives in interdisciplinary data science research and education. In May 2023, she began a new role as computational biology lead at Avalo AI, where she's continued promoting feedback between machine learning, evolutionary genomics, spatial ecology, and plant and data science toward more resilient and sustainable food systems. Emily, it's a pleasure to have you here with us, and I will stop sharing here for you to share your presentation.
All right. Thank you so much, Carlos, for the introduction, and thank you to everyone for joining today. I'm going to be talking about how we can use knowledge of evolution to accelerate breeding for current and future environments. And I wanted to start with one of the core questions that's driven a lot of my research for many years, which is how can we unlock big data, so high dimensional multimodal data sets to understand how plants adapt to environment, with the idea that if we can understand how plants adapt to stresses, drought stress or biotic stressors, things like that, then this knowledge can enable us to develop more resilient crop varieties. And I've been really lucky to start working recently full-time with Avalo. So if you don't know this company, we're a small company based out of Durham, North Carolina. And our mission is explicitly focused around using evolution and advanced computational methods to accelerate crop improvement. So really lucky to find a company that shares my two obsessions there. And this has been a really interesting experience and a sort of rubber meets the road kind of moment for me personally, as I've started to really think about how do we use knowledge of evolution in decision-making for a breeding program. So, it was always something that I talked about as an academic in my grants and my papers. If we understand the genetic basis, yes, it can help with breeding, but now I really come to appreciate, and I think many of you all will also appreciate, it's not always that simple. So that's, I think, the question I wanted to focus around for my talk today. How does understanding evolution inform breeding? How do we incorporate knowledge about evolution into breeding programs? And that's a question I hope we can have a really active discussion about at the end of my seminar today. And I'll share a few perspectives from my own work in collaboration with many wonderful colleagues in Striga and sorghum system, and as well in rice. All right, so I want to share first one of our bread and butter methods for developing hypotheses to understand the genetic basis of evolution in diverse crop land races. And this is genotype environment association analysis, or GEA, as I'll call it for short. And the general idea here is if we have access to really incredible genetic and germplasm resources, like we do for sorghum, for example. So what I'm showing you in this map here is more than 2,000 sorghum land races. Each of those point is a separate land race distributed across many different countries in Africa. And they're colored actually by the level of rainfall at that location. And so the idea here with a GEA is that land races, because they've been passed along for many, many generations, if we hypothesize that they're adapted to their environment of origin, if we find alleles that are more common, for example, in a very dry in very dry environments, those alleles might be important for adaptation to drought, for example. So that's the kind of signature that we're often looking for with a GEA analysis. And in practice, this is basically a GWAS, where instead of a phenotype, you have environment like precipitation at the location of origin for your dependent variable. All right, and when I first started using and developing genotype environment association frameworks for thinking about evolution in crop land races, actually one of the first questions that I had, and this is work in collaboration with Jesse Lasky at Penn State, was what about biotic environmental gradients? So can we extend these powerful GEA frameworks to pest and pathogen systems to, for example, identify alleles important for tolerance or resistance to parasites? And it's a little bit more complicated than it is for abiotic environmental gradients. For example, there is a lot of environmental variation, of course, but a location near a desert is always going to be relatively dry compared to, for example, a location near rainforest. But within populations that are exposed to parasite pressure, the parasite can change a lot within that same location. So we weren't totally sure when we set out to look at this question if this type of framework would be suitable for biotic environmental ingredients. And we set out to test this with a really important and devastating host parasite system. So this figure here is showing you this pink flower in the front is a Striga hermanthica. That's a parasitic weed that is considered one of the greatest biotic constraints to food security in Africa. And I've got some stats here. These are woefully out of date at this point in terms of the number of people and economic losses impacted. It's also a good model system because we actually know a fair amount about the genetic basis of resistance mechanisms in this system. So very interesting biology, actually. The hosts emit strigolactones, chemicals from their roots, and these actually stimulate germination by the parasite. These parasites actually adapted to eavesdrop on these strigolactones, which are actually meant for to stimulate beneficial interactions with mycorrhizae. So especially under low nutrient, low water conditions, diverse strigolactone molecules are exuded by host roots in order to recruit and stimulate interactions with mycorrhizae, and then the parasites cue in on that signal. That stimulates their germination, and then they continue to develop a special organ called a hostorium, which infects the host root. And through that connection, they establish vascular connections and can siphon water and nutrients from the host. In 2017, there was an excellent paper in PNAS which established LGS1 as a particular locus, major effector locus, which causes a change in the chemical structure of one of these strigolactone molecules that's associated with reduced infection by the parasite. So the parasite, or some populations of the parasite, I'll qualify, do not germinate at as high frequency when they detect the left-handed form of this molecule, whereas 5-deoxystrigol is a much more potent stimulator of germination. And I want to fast forward a few years because I think this has been a really important discovery that's enabled the development now of, we'll call it Striga Smart Sorghum is the tagline that my colleague is using for this, these Striga Smart varieties. But essentially, we're able to now use gene editing to take farmer-preferred varieties and introgress this loss of function in LGS1 mutation. So this is pretty exciting development now to be able to deploy many of these more striga-resistant sorghum varieties. And I want to also acknowledge a lot of the work of a collaborator, Dr. Stephen Runo, who really helped, I think, pave the way deployment and development of these varieties through a lot of the work of his lab and the lab of others. So here we are now, a really exciting moment for Striga Smart Sorghum. But I just wanted to highlight this as, this is a situation where this LGS1 location in the genome is also like a really great locus that we can use for validation of new approaches to look for genes important for parasite resistance. So we did that actually using a new method based on ecological niche models. So one of the challenges with biotic gradients is, unlike abiotic gradients, where we can kind of go to find publicly available data at high resolution for things like annual rainfall and temperature related variables, we don't often have gridded high resolution data for parasite occurrence. So we developed a method to generate those kinds of maps using habitat suitability modeling with MaxEnt. So the idea here, essentially, this is a machine learning method where we can take information about species occurrence for Striga because it's very well represented in herbaria as a plant, as a parasitic plant. That's where we are able to get a lot of good occurrence data from. And then what we can do is look at values of the environment where we see the species occur, and then generate predictions of where we expect the parasites to be likely to be based on values of the environment at known locations of occurrence. All right, and that's what we can use actually as our dependent variable now in a GEA, and that's what this GWAS is showing you, is across the genome associations with modeled parasite occurrence score. And what we can see is that we have a few locations that co-localize with known QTL for different Striga resistance mechanisms, as well as known genes involved in the strigolactone biosynthesis pathway. Conspicuously absent from this plot actually is that locus I mentioned before, LGS1. And there's actually several reasons for that. One reason is there's quite a lot of diversity actually at this locus. So multiple LGS1 variants are associated with Striga occurrence. So if we split those out separately, we actually do find strong associations with those individual loss of function alleles. Another reason is that a lot of the variation at that locus is structural. So if we use kind of normal SNP-based approaches, we will lose information about sites where we just don't have a call, because those are large loss of function. Several of the loss of function variants are quite large. Another reason we don't find a strong signature in the first pass GEA is this signatures of association with environment are also confounded by population structure. So that's kind of a limitation that can reduce power in GEAs. So several different reasons why we actually don't see this locus pop up in kind of a GWAS with parasite environment. Although if we look closer, we do find these associations, and we were lucky to be able to follow up at this region and look at other signatures of selection. We had colleagues did some CRISPR editing, and so we're able also to show that this does have a benefit, also slight negative effect in terms of growth rate if you knock this gene out. But all that to say is that, coming back to this question of how does understanding evolution inform breeding, one of the big ways is through gene discovery, right? So if we can find genes important for adaptation to environment, that can be really valuable. Those could be really valuable targets for plant breeding. But it can be really complicated. I think that's one of the major lessons I took away from this work with Striga, is that we can miss a lot of really important genes. And a lot of times the traits that we are interested in have a very complex genetic basis. So I've become very interested more recently in how we use knowledge about evolution and kind of the flip side of the coin, genomic prediction, right? So I'll talk a little bit about some ways that we've thought about incorporating evolutionary information into genomic prediction problem. And for that, I'll talk a little bit and switch systems a bit into rice. So rice is, of course, a really critical staple food crop. It's also a crop that has outsized impacts on the environment, and this is because it's a semi-aquatic plant, so it's often grown under continuous irrigation. Because of that, this anoxic soil environment can create conditions conducive to emission of methane. It's about estimated 12% of anthropogenically produced methane is from grass or from rice production alone. So that's 1.5% of total greenhouse gas emissions, and that also impacts for irrigation a lot of the causes a lot of freshwater withdrawal. So there are many methods that have been developed to try to decrease the amount of water that's used to grow rice. Furrow irrigation, alternate wetting, drying, things like that. But there can be a challenge, especially if there's excess nitrogen around, in that these dry downs of the soil can promote emission of nitrous oxide, which is even more harmful greenhouse gas actually than methane and carbon dioxide. All right, so challenging balance there of how do we produce this critically important crop, but also reduce the amount of water and nutrients that we're using. So rice, of course, a very globally important crop, also very locally important to my home state of Arkansas. So I can't get away without showing this map. So in Arkansas in the US, we produce about half of the rice that's produced in the US. And this also has a big impact on the water use in our region. So about half of groundwater use in Arkansas is for rice irrigation, and groundwater levels in this area are also declining. And so I put a pink star here. That's where I'm from, right in the middle of this rice growing region. Actually, that is where Arkansas State University is as well. So really perfect area to be thinking about rice research. But as we think about how can we mitigate some of these challenges, is there information about the evolution of the species that could be helpful? One of the things I think we can think about is the really interesting evolutionary history of this species. So rice was primarily domesticated within irrigated environments. So this is a map of the dispersal of Japonica rice. So Japonica was domesticated in the Yangtze Valley about 9,000 years ago, and then diversified into temperate and tropical Japonica about 4,200 years ago before spreading into Southeast Asia. And then here's a similar map for indica rice that was domesticated in the Ganges Valley around 4,000 years ago, and then spread into China around 2,000 yeast ago. But through all this dispersal through to new environments and new growing conditions, rice landraces had the opportunity to adapt to many different kinds of growing conditions. So there are a lot of different irrigation systems that landraces can be found in. So from irrigated environments to upland environments and rain-fed lowland environments that experience a lot of intermittent drought. And then there's this really interesting deep water and flow tank cultivation systems as well. So all these other kinds of cultivation systems where landraces would be expected to experience more drought might be good sources to identify alleles important for adaptation to dry land conditions. And there's some evidence that a lot of the selection across the rice genome as landraces adapted to these environments is actually to promote and intensify interactions with mycorrhizae, which is also really interesting. All right, so here's our question. What is the genetic basis of rice adaptation to drought-prone environments? And then the other second side of this question, can we use this information to accelerate development of nutrient and water efficient varieties? When we first approached this question, here again, we are using environmental niche models as a way to reduce dimensionality of environment and basically capture, I think, irrigation system as an environmental gradient. So the way that we did this was by treating landrace presence in a rain-fed lowland environment as a presence, and then we consider environments that are enriched for high habitat suitability for rain-fed lowland environments. And that's what you're looking at in this map here. So this is a map of habitat suitability for rain-fed lowland growing systems. All right, and those are based on, as input, 8 environmental layers. So we have soil and climate layers in this case, and then the habitat suitability models are constructed with MaxEnt. We then, just as we did for striga and sorghum, carry out a genotype environment association analysis where the predicted habitat suitability for rain-fed lowland environment is our response variable, and this is using a fantastic data set of more than 1,000 rice accessions And then we've got a subset of SNPs here that overlap with one of the panels that we are looking at for validating some of these loci in experiments. And I just wanted to share with you a little bit about what the rain-fed lowland, and actually here's also the habitat suitability models for upland varieties look like as well, and why I like them so much as a dimensionality reduction technique for environments. And the reason for this is because these environmental niche models or ecological niche models do a really good job of capturing nonlinear relationships between species presence and environment, and also in weighting those variables appropriately. So that's kind of in contrast to many other multivariate approaches for genotype environment association analyses. And so just to show you here for rain-fed lowland, that was growing degree days was the most important variable for that habitat suitability model. And you can see higher habitat suitability as growing degree days increase. Max temperature of the warmest month, we're using that to capture heat stress as it gets hotter. Habitat suitability decreases. We've got growing season precipitation, and then soil pH was the fourth most important variable, actually for both of these models. And moving forward for GEAs, we actually end up just using the rain-fed lowland model, mostly because they look pretty much the same, except for at the ends of the values for each of these input variables here. And I think that's just because we have a small input data set for training. So there's a little bit of inconsistency there. And then here I'm just showing you kind of sanity check figure where we have the habitat suitability score on the x-axis for both of these models, and then the density for across the training set of accessions that are from different irrigation systems. And actually what we see is that the rain-fed lowland models, for example, actually tend to overlap a lot of the areas for some of the other dryland irrigation systems, at least in terms of the projected habitat suitability. So we have upland varieties actually end up having pretty high habitat suitability score for rain-fed lowland locations, and also the deep water have pretty high habitat suitability there too, which is very interesting to see because the deep water rice often, in addition to flooding tolerance, they often, many of those accessions will have high drought tolerance as well. All right, so this is what our GEA looks like. You could certainly use GEMMA or another kind of method. I'm actually showing you a figure that for calculating the actual associations, we're using Avalo's method for linking position in the genome to environment. This is GDIP, or gene discovery through information-less perturbations. And so it looks Manhattan plot-like, but the distinction here is that this method, each position indicates a SNP in the rice genome, but the association, this association statistic tells you about unique information that's explained by that SNP compared to many others. So I think we're excited to use that method compared to kind of traditional GWAS approach. I want to thank Mariano here. He's a co-founder and CSO of Avalo and developed this for this algorithm. And then Emily Abernathy too is our bioinformatician who helped process a lot of these data. All right, so what can we do with these environment-associated alleles once we find them? And I said that we were interested in genomic prediction frameworks. So one thing that we've looked at is whether environment-associated alleles can help improve genomic prediction models. So the idea here in our hypothesis is that if we have well-chosen subset of features, so we have much fewer features, This enables training of more complex machine learning models because with fewer features, we're able to learn nonlinear relationships and more complex interactions more easily than if we have kind of the typical amount of SNPs that you would have in a genome. So that can make learning these more complex interactions more challenging due to this cursive dimensionality problem that we often face in plant breeding, where we have far fewer samples than we do predictive features. And to test this, we are using published data from Niels Gruen from a 2020 paper. This is an experiment they did where they have 206 diverse rice accessions. They're grown in drought and well-watered conditions in the Philippines. And they measured several different phenotypes. I'm just looking at grain fill here under drought conditions, and we're comparing machine learning models versus our BLEP as the benchmark. All right, and here's what our results look like. And I think as well, Justin, our machine learning specialist at Avalo, for training these models. And what we find is that our approach does use far fewer sites, so around 7,000 compared to 173,000. So that's after a little bit of light LD pruning. And we actually get pretty similar results in terms of predictive performance for this grain fill under drought in the Philippines data set. So I guess our model is slightly better. It's very subtle difference, and we're able to create good predictions with fewer SNPs, which can be useful under some contexts. Although I have to say, these are pretty new results. I was hoping for a little bit larger of an increase. We often will see much higher improvement compared to our BLEP with some of the more traditional GWAS data sets that we work with. So I kind of had been wondering, well, are there any other ways we can increase predictive performance with this subset of evolutionary informed SNPs? So one question was, could there be issues with generalization from our feature selection set, which is the 3,000 rice genomes landraces, to this data set that we actually have phenotype data for in Gruen et al. I actually don't think that's the case. I think a lot of diverse rice germplasm is well represented in both data sets, as you can see from this figure. This is a PCA. And we also have, I've also put the USDA rice mini-core there. So the USDA mini-core germplasm is hosted in just down the road from Jonesboro, Arkansas in Stuttgart. And so this is easy access to us for experiments. We can see that there is, this one represents a little subset of that diversity that we use for training the models. What I really think actually could help us here, though, is higher resolution in the phenotypes that we're looking at. So this is summary data for the phenotype that we used for training. These, again, are data from the Gruen paper under wet and dry conditions. And you can see under drought, because of that strong selection, the values that we're looking at are ranging really from 0 to 500 or so. And that's really just one dimension of phenotype, right? And really, phenotypes are very high dimensional, varying over time, and especially with the ability to collect image data. I think just looking at grain fill is perhaps a poor reflection of everything that's going on in the plant and everything that our drought-associated SNPs could be impacting. So the next thing that we are going to be looking at is whether evolution or evolutionary informed feature selection can help prediction models for more complex phenotypes. So here's our question: can multispectral, multitemporal phenotypes provide a more complex view, more comprehensive view of complex trait variation? We haven't really, we're just kind of getting started. This is a new project funded by the USDA Data Science for Food and Ag Systems program, and that's just started in August of this year, and this is in collaboration with Dr. Halia Lawrence at Arkansas State University. So I don't have any data to share with you about that yet, but I do have some results from an ongoing project, my former master's student at Arkansas State University, that's looking at drone data instead as a way to capture complex phenotype information. So this is data from a field season where we have about 12 flyovers during the season. The spatial resolution is about 2 centimeters, and we've got a multispectral camera plus thermal, so 6 bands. This project also is in collaboration with Ahmed Hashem at Arkansas State University. So he is really the person to thank for the fantastic drone image data set that we're using for this analysis. And the way that we've been thinking about these complex multitemporal phenotypes, one of the approaches that we were really excited to try on these data is kind of borrowing approaches from the single cell genomic sequencing community that they've used for trajectory inference, where you have development, even when you measure something at a particular time point, you have a lot of variation in developmental stage across many of the cells, and that's kind of the same problem that we face when we're looking at drone images of a variety trial. So I'll tell you a little bit about one of the approaches that we are using. It's called manifold learning. The outcome is UMAP, in case anyone is familiar with that, but you can think about it as a nonlinear dimensionality reduction approach. The idea here being that often data represented in high dimensions are really sampled from lower dimensional manifolds. And I've, in my image here, I want you to imagine a Swiss roll with a bunch of rainbow sprinkles on it. And, you know, we bake it in a kind of 2-dimensional pan, and then we roll it up. And we have this kind of roll shape here. But in reality, this comes from a 2D piece of cake, right? So if we were to apply something like PCA, which may be the dimensionality approach that people are most familiar with, what happens is we basically would just smash the Swiss roll, and we would have the situation where, for example, purple sprinkles are pretty close to sprinkles that they're actually not that close to on the lower dimensional manifold. But other approaches like UMAP have the potential to unroll the Swiss roll, essentially. And now regions that are close together on the lower dimensional space end up in our lower dimensional representation close together as well. All right, so we were interested in applying this approach to the problem of drone data for rice, and the study that we used to look at this is a study that was carried out at the USDA National Rice Research Center in Stuttgart. And this is kind of an aerial view. We've got 2 years of data, so 2021 and 2022. And there's several different mini experiments going on in this study. So we've got a few variety trials where we have different nitrogen rates and seeding rates. In 2022, we just did one seeding rate because we had pretty similar spectral signatures for the seeding rates in 2021. We also had a hybrid inbred study, a few accessions from the mini core, and then our colleagues at USDA collected a lot of phenotype data for ground truthing, including days to heading, percent lodging, leaf temperature, SPAD yield, and many other things. I just kind of want to show you an idea of what the yield variation looks like across these 2 years and for several of the different genotypes from the nitrogen rate study. And I'm highlighting, so this is nitrogen rate on the X, so that's 3 different nitrogen rates, and then on the Y is yield. And then I've highlighted in pink a nitrogen sensitive variety, that's Roy J, and then a relatively nitrogen insensitive variety, Santee Gold, where yield does not change in response to increased nitrogen addition. And then here is what vegetation indices look like for, here's the nitrogen sensitive cultivar. So this is day of year on the x-axis, and then just one spectral index on the y, so that's NDVI. And then we've got some replicate plots. In the dash line is low nitrogen for Roy J in pink, and then the solid line is high nitrogen. So you can see quite a big difference in this nitrogen sensitive variety, especially in 2021. And then here is our nitrogen insensitive cultivar. And so for this one, we definitely can see some distinction in the vegetation indices that we don't see in the yield data, and especially early in the season, but especially in 2021, not so much in 2022. But this is really just one index of many. So our question is, okay, if we can capture many different indices or many different channels, can this help us in capturing trajectories or stress response trajectories? And I do want to emphasize that we're kind of creating this high dimension problem for ourselves in a way, because we have, we're starting with 6 features, 6 bands. Then Fareed created many derived features. So he's got vegetation indices here and also image texture features. And then our goal is to reduce these again back into fewer UMAP dimensions. But we are working on, Fareed is also working on parametric UMAP approaches where we take the images and embed them directly into the latent space. All right, so here's what some of the UMAP embeddings look like. This is still from the 49 vegetation indices. So here's for training. Each point in this figure represents a different rice plot at a different time point. So you can see there's definitely a trajectory here from early planting to late planting. That's how we train the embedding on that 2021 data. And then in 2022, this is all of the accessions are projected into that same space based on their calculated vegetation index and texture index features. All right, so again, kind of this progression from early to late here. And I also wanted to share with you, highlight specifically these nitrogen sensitive and insensitive varieties. So this is, each of these 5 panels is a different day after planting. But what you can see, and I think this is especially apparent day 102 here and day 82, is that for nitrogen insensitive variety in blue, these there's certainly a lot of variation, but these points are much closer together for the nitrogen, low and high nitrogen, than they are for the nitrogen sensitive variety. So I think that these latent spaces are actually doing a pretty good job of capturing variation in stress response trajectories in this space. The last thing I want to show you is this figure, which is a bit busy, but the main takeaway is that when we carry out these UMAP transformations and then try to predict something like yield from coordinates in this latent space, we actually have better predictive accuracy than we do if we just use the raw vegetation index features. So I've just got in purple, this is the results for a couple XGBoost models. The first one is based on the UMAP features. The second one is just based on the raw features. So lower mean absolute error for UMAP transformed inputs. And then LASSO is a different type of model architecture. And again, we see improvement with UMAP features compared to raw. And Fareed has since carried out some more modeling with a bunch of other kinds of architectures. And so far, the UMAP transform features are, these results are holding across other architectures as well. All right. So just to end with a few conclusions here, how does evolution inform breeding? Well, for gene discovery, signatures of selection can help identify gene targets, but I think it's complicated. Structural genomic variation, correction for population structure, diverse alleles can really mask the signal and make it difficult to find alleles if you don't already know something about the genetic basis of them. So we have some evidence for that from the Striga sorghum system I talked about. But I am very enthusiastic about how knowledge of evolution can be used for genomic prediction. So we saw that evolution-informed feature selection can be useful for genomic prediction models. So in the kind of simple trait sense, so we have a slight increase in performance for evolution associated features compared to many features across the genome. However, I think the real promise here is in considering an expanded view of breeding targets. So rather than a few traits like, of course, yield is very important, but thinking about traits as kind of a simple, few traits, thinking about really more expanded view in higher dimensions. So breeding for latent space trajectories, for example, breeding for ecological niche, I think it's really promising area for the community. So I gave some example for latent space trajectories. For ecological niche, we are actually starting some work in that soon as part of a new project. I'm shamelessly calling this Avalo gem after our company, accelerating variety adaptation to local environments. And so we're kind of starting this work in rice, but if anyone is interested in other crops or just wants to talk about what we're thinking about there, please feel free to get in touch. And with that, I'd like to thank many people and wonderful collaborators on the work I shared today, as well as support from the USDA, from an NSF grant, and from Arkansas State University and University of Arkansas Division of Ag as well. That's helped get a lot of this work started.
Thank you, Emily. Thank you so much for such an interesting work. Again, the questions, please post the questions in the chat and we relay them to Emily, or Emily, you can read them directly. Listening to your work in sorghum with Striga, I wonder what is the capacity of the parasitic weed to evolve simultaneously?
It is really, they evolve really fast. So that's actually something that Steve and I have talked a lot about, because there is, we also see a lot of evidence for variation among Striga populations and the ability to overcome that LGS1 mediated resistance in sorghum. So actually where he is based in Kenya, those tend to be like pretty virulent Striga populations. The germination is still reduced, but it's much higher than, for example, populations in West Africa. That LGS1 mediated resistance is much more effective. So a lot of variation within populations. The Striga hermontheca is obligately outcrossing. And so, you know, I think it would be fantastic if sort of trying to guard against evolution of the parasite was part of the deployment strategy, and it's definitely something that's on people's minds. But I think perhaps it also is only a matter of time until many populations could adapt to overcome that resistance.
Is that gene deployed already in the field?
Not the CRISPR edited lines to my knowledge, but it's pretty close.
Okay.
So.
Any questions for Emily, please type them in the chat. The presentation was so clear, Emily, or we didn't understand anything.
It might be more the latter, but maybe I posed the question myself to the group, is if you are a breeder or work in this area, how do you use evolution in your work?
Yeah, that's a good question, and probably this is like a ping-pong match. I'll turn it back to you. Given the rapid evolution of environments that we're having, how do you think your work can help us develop better resilient materials?
Yeah, so maybe I think the last slide I showed is maybe a little teaser for that. And the maps there, you can think of them as, the ideal thing would be if we had an input genotype and we can predict the suitability of that genotype across many locations. I think the goal of that work will be to enable us to breed for varieties that do very well in a particular local environment, as well as varieties that do very well across a broad range of environments. So that, I think, is the vision.
Right. Let's give a few more minutes to see if we have any questions. We're gonna post the recording tomorrow with the announcement for the seminar next week. There will be a link for the recording to this seminar in case you wanna review it or share it with somebody else. Let me share this screen again. Because in the meantime, if we receive any questions, we'll go back to you, Emily. But thanks for the attendance, everybody, and thanks to Brandon and Thiago for their help in organizing and making this seminar series a reality. Next Thursday at the same time at 11 a.m. we're going to have Dr. Hamida Shafri from our university talking about blueberry breeding and sustaining the supply of high quality blueberries by leveraging applied breeding genetics and genomics. Okay. And we look forward to have you here again. Thank you so much. Thank you, Emily, again for your wonderful presentation. And it's great to have you with us. And there's a question now. It is possible to identify parasite resistance gene to improve sorghum?
Yes, and there has been a lot of work with kind of GWAS based approaches where you can do parasite pressure and not. And I think that's been very valuable and important work as well. I think a lot of the motivation of our GEA-based approach is, can we find different genes through this other, I would say maybe easier method where you don't have to go through all of the work of the variety trial in a way to kind of complement the GWAS-based approaches that many have done for many different parasite populations and across many locations as well.
I was wondering if Striga is the main parasitic weed that we know of. Are there other examples, and what learnings can be?
Yeah, so there, even Orobanchaceae is the family that includes Striga. There are many diverse species of parasitic plants, including ones that are not weeds. So mistletoe, dodder includes many both weedy and non-weedy species, and as well as many agricultural parasites. But in Africa, especially, I think Striga is the main main one.
So there's a comment here, a question he wants is In our work, we identify many strains of Striga with completely different virulence. How can we use genome editing to edit the virulence?
Yeah, that's a difficult, it's a difficult challenge, right? So I think even what we see as well with LGS1 is that we can edit the host, and we are going to have very varied effects across Striga populations that across many different countries of Africa. There have been suggestions for host-induced gene silencing, which I think is still very much still in research phases for the system, where you could edit the host, and then the attachment already has to form in order for genes that you edit in the host to be sent over typically. So I don't know, I think we're still a ways out with Higgs for that to be an actual effective strategy.
Okay. Yeah, it looks like Striga has a breeding program of its own, and it's very efficient, I would say effective. Mohammed says, I'm working on the CCO gene family in rice by genome-wide identification, which relationship with stress. I don't know if it's a question or it's an assertion.
Yeah, I haven't looked into any of the hits that we've found yet, but I'll keep an eye out to see if we see any links to CCO gene family. So thank you for your
lead. Yeah. Well, thank you so much for your questions, and thank you so much for your time, Emily. Appreciate it very much. And you guys have a very nice rest of the week and a good weekend, and I'll see you next Thursday.
Thank you.
All right. Bye-bye.